We have a DNA sequencing machine at the Creation Research Society. We're going to find out what that's going to be used for. Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
And your co-host Rich Gear here as well. And Doug, I'm really excited about our guest that we have tonight. You want to introduce him? I'm really excited because you're a big part of this.
Okay, and this is Dr. Joel Brown. He is
Yes.
the director of the Andel Creation Research Center in Glendale, Arizona. And this is at the campus of the Arizona Christian University. And the Creation Research Society is, I think, the oldest creation organization that is still continuously operating in the world. Am I correct about that, Joel?
Yeah, you just took all the words right out of my mouth. I don't have anything to say now. You're spot on. So we bill it as the oldest and the largest network of creation scientists in the world. Founded in 1963, a little before my time, and it has continued to this day.
Was this after because the Genesis flood was written in 61? Was this came in after that then? Am I getting the time frame right?
Yeah, so Henry Morris was one of the founding members of the Creation
Okay.
Research Society. And then he also went on just a couple years after to found Institute for Creation
I
Research.
see. Oh, okay. I didn't realize he had done both of those.
Yeah.
Okay, great.
And I mean, it's helpful then to know kind of the difference, the way CRS is designed to function. We're designed to be like an umbrella organization, an umbrella network that connects all creation scientists. Because some creation scientists are at Answers in Genesis, some are at Creation Ministries International, some are at Institute for Creation Research, and many are just independent researchers at universities,
Sure.
and so the society is meant to connect all of those.
Mm.
So you have recently acquired a new piece of equipment. Can you tell us a little bit about it?
Yeah. Okay. So, we, my desire ever since my PhD is in molecular biology and genetics from Cornell University. And we studied developmental biology, how organisms develop. And of course, that involves DNA. So, really, I have a genetics background. I'm a geneticist. And only in the last couple decades has sequencing the DNA at a large scale become feasible. And even though it was feasible, the ability to sequence DNA was always housed at large facilities with multi-million dollar machines and operations.
Yeah.
And so you would send your sample off-site to a facility. They would sequence it for you, send you back the results. And only in the last 10 years, the technology has progressed to the point that now you can sequence billions and billions of letters of DNA right in a lab like our own. And so this past summer, we had the honor to be able to purchase one of these with the help of Doug Sharp and Revolution Against Evolution. We purchased a sequencer. Now, these things are about the size of an eraser or about the size of your smartphone. And that's considerably smaller than they used to be. And the technology now that they use to sequence is quite amazing.
It's kind of like computers in general. Remember, they used to be the big Fortrans or whatever they were, took up whole rooms, and now the size of a cell phone, you can just you can do what you can do more complicated things than they did in all those big machines. And I guess same thing worked with the DNA sequencer as well, right?
Yeah,
What's
yeah.
exciting about? Go ahead and go ahead.
Let me see here. I there it is. I actually brought one of the inserts from the lab. So this is the sequencer.
That's as big as it is now, huh?
Yeah,
And that
now
no, I'm looking at it, but I'm just amazed that it's that small and used to be really huge though, right?
yeah, and they still have the really huge ones. This is a new technology. Um, it's called nanopore sequencing, Oxford Nanopore Technologies. I'm going to get it to focus again. There it is.
There you go. That's good.
And so you inject your DNA over here on this side and it flows through. And of course you can't see the DNA. It's microscopic, tiny molecules. You can't see them. It's just in solution. It looks like you're injecting water. And then the DNA flows to this sensor area, and that's where the magic happens. Now, of course, this is plugged into a separate device that connects it to a computer, and the computer's doing the heavy lifting. Let me just kind of give you a brief idea of how this technology works. So you have, imagine a membrane, a very thin membrane, and you have all of these pores in it, all of these holes. These holes are made out of proteins, so they're tiny microscopic pores in this membrane. And you've got your DNA on one side of the membrane, and it's trying to cross over to the other side of the membrane. So you get these little carrier molecules, they take the DNA, they hook it to the pore, and this is all happening at a microscopic level. There are 2,000 pores in this sensor. And so the strands of DNA will bind to the pore and they will begin to get threaded through that pore. And as it's threading through the pore, it's sending an electrical signal in that pore. And the electrical signal changes depending on which base, which letter is passing through at that moment, whether it's an A, a T, a C, or a G. And so you get a pattern, like a spectrogram of this electrical signal and how it's moving up and down. And then the computer interprets that. That's called a squiggle. That's the technical term.
Wow.
Called a squiggle. And the computer interprets that electrical charge and the change, and it reads it as a letter in the DNA. And it's doing this, 2,000 pores simultaneously. So you've got 2,000. That's best case scenario. Let's say 1,000 realistically, 1,000 DNA sequences running through 1,000 different pores all simultaneously, and the chip is detecting all those electrical signals and interpreting the sequence.
Man.
It's crazy. I mean, to think that this technology has progressed to where it is now, it's amazing.
One thing I was thinking about is a possible experiment. And you were doing some things with cactus, but I was thinking of taking the two different uh, things people attribute to convergent evolution, agave and aloes, and comparing the DNA sequences of both of those and see if there's any design similarities or differences, because according to the evolution, because they are different, come from different parts of the world, they are not related, and yet they have similar characteristics. So it might be an interesting possibility. Now, I have your video here that we can show, which,
So while you're queuing that up, let me kind of explain to viewers the two things that we do in our lab.
yeah,
So our lab is here at Creation Research Society in Arizona, and our lab is equipped to do two things that we focus on. Number one is molecular paleontology. So paleontology is the study of fossils in the ground. Molecular paleontology is the study of molecules in fossils in the ground. So we study molecular paleontology, and the other thing that we do is we study, the fancy term is phylogenetics. It's the genetic history of species. And from a biblical perspective, we're studying the genetic history of species and how they developed from the flood. So those are the two things we focus on. If that video that you're gonna pull up, that is us studying cacti. And so we wanna understand, like, what did God create the saguaro cactus and the mistletoe cactus and the prickly pear cactus as all three separate creations, or are cacti all related, or are
Oh,
they in two separate groups? And so one of the ways we can do that is by looking at their DNA. So that was the first project that we used the sequencer from, and it's ongoing. We got our first sequence just in June off the sequencing device. It was a
can you tell us any conclusions or any thoughts yet or not?
well, I can tell you what we from this particular sequence, we have not compared it to the other sequences that are
Okay.
out there. But based on other sequences that are out there, it looks like cacti form two groups. The prickly pear is one group, and then the columnar type are another group.
Oh,
And
so,
so
oh, okay.
that's what we're trying to decipher.
Interesting. Okay. Wow. Well, you were talking earlier before we got on camera there, and I don't know what I'm jumping the gun on this, but I'm fascinated that you're able to get some DNA sequencing out of actual fossils. And I thought, I knew that there's been hemoglobin and soft tissue and all kinds of things. And I figured you could get some DNA from that, but that you could actually get it out of mineralized bones or something. That fascinates me. I did not realize that was even possible. And you're saying it is possible, or am I misunderstanding?
No. So what is happening? I mean, during what you're talking about is called taphonomy. That's the fancy scientific word
Okay,
for the process whereby something living becomes um, lithified. It turns into a rock, becomes
right,
fossilized. And so the minerals get replaced by geological minerals, and the proteins and other biological molecules they break down and degrade and are washed out. Um, but if the conditions are right, then that DNA is just DNA will sit in dirt. It'll sit in rock. It'll just hang around. DNA is very fragile though. So it's not nearly as stable as proteins, which is why you hear the stories about proteins that are being found in fossils. Those are a lot more, a lot easier to find. The DNA in a dinosaur fossil is going to be very, very difficult to find, but we believe it's out there because we believe dinosaurs died off during the flood a mere 4,500 years ago, not 66 million years ago.
right. Okay. So have they found any yet, or have they found some DNA in any of these fossils, or because I know
Well,
there's some of these like hadrosaur skeletons up in Canada that have the bones have not been mineralized, as I understand it. They're unfossilized, and that it seems like you can find some DNA a little
that
easier. Yeah, go
That
ahead.
is exactly where we're headed. So I think this is going to depend a lot on the conditions in which these bones are deposited and then preserved. So in cold environments, you're going to have much better preservation or much higher likelihood of DNA being preserved, as well as very dry environments. So those are two possibilities, and that's so here. Let me kind of tell you where we are, the state of the field right now in this ancient DNA research. There are about four different creation labs that are working on this, and there are several secular labs that are working on this.
Mhm.
In the secular world, they've been able to get DNA from mammoths, and they say that's 1.5 million year old DNA. So we do, and of course, we would put mammoths post flood, so we're talking about 4,000 year old
Yeah,
DNA.
yeah.
That's still really close to the flood timeline. And so, based on that, we would expect we could extend just a few hundred years back and be able to get the DNA from dinosaurs. The conditions are different. These mammoths are frozen. They're well preserved, and so we're going to have to find equally well preserved dinosaur fossils.
It's interesting that at first they said this wasn't possible to find DNA or proteins from something millions of years old. But now that it's becoming commonplace in the research, oh, by the way, yes, it can last millions of years. They don't question the assumptions behind it.
Well, have you ever heard the story of the dead man that bled? Let me tell
No,
you
I
this.
do not know that story. Tell us the story.
So, the man shows up to the doctor's office and he says, "Doctor, I have a problem. I'm dead." And the doctor said, "Well, you're here. You're not actually dead." And he goes, "No, no, I'm dead. Prove that I'm not dead." So the doctor said, "Okay. Do dead men bleed?" And the man said, "No." So the doctor took a little needle and pricked the fella, and he started bleeding. And the man said, "Well, I'll be. Dead men do bleed."
Yeah. Okay. I knew where that was going once you started going. That's exactly what they're doing with
Yes.
the dinosaur stuff. It's like, okay, no way could DNA or hemoglobin or collagen or any of these things possibly exist in 66 million, let alone 100 million year old, 1 million year old. But now all of a sudden they're finding it. And I think they're finding it. Well, I got the what's her name, who found the hemoglobin? I always forget her
Schweitzer.
name. Schweitzer, yeah. And she was an honest evolutionist, I would say, because she
Yep.
admitted, they found it. But outside of that, they never looked for it. It
Well,
was the creation. It was the creationists that were willing to look for this stuff.
yeah,
So
I mean,
it's
she actually took a lot of heat for
yeah,
early on for proposing this preposterous idea that proteins could still exist in these 66 million year old tissues. And so she has defended that, defended it. And I would say it took about 20 years. And now the science, mainstream science, has fully embraced it and accepted it. There are a few outliers that still say it's exogenous protein that's gotten in there, but they've stream science has accepted it. And now they're saying yes, so proteins do persist, but there's no way DNA persists. So they're kind of doing the same, playing the same trick on themselves again. We believe that they're kind of have a blind spot because they're so insistent that these things are 66 million years old that of course DNA can't last that long. Proteins, maybe. They've changed their mind on
okay.
that, but DNA, no way. So it's a really good opportunity for us as creationists to kind of stick our neck out there and say, look, we would predict that DNA is still in these fossils. Not all of them. DNA is very fragile. It's not going to last long, but under the right conditions, we should be able to sequence dinosaur DNA.
Share your multimedia presentations here, Joel, and we can have you narrate them.
Absolutely. Go ahead. And to kind of cue this up, so this is going to show the story of the first sequence that we got from our lab.
Well, there you have your lab crew.
Yeah, that's part of the crew. You won't actually see them in this particular video. Okay, so I'll kind of talk through it as we're going. Can you hit the full screen there in the bottom right? Yeah, right there. All right, so these are different. This is a teddy bear,
Hey,
Choya.
hey,
We went to private property with permission and collected all these different cacti. So that
okay.
was step number one, sample collection. Now these cacti are filled with DNA, and we are actually trying to sequence the chloroplast DNA. So there's the nuclear DNA and there's the chloroplast DNA. So we are kind of scraping away the tissue. The green tissue is filled with chloroplasts. And then we're making these cacti smoothies. So this is a mixture of all the cellular debris from the cactus along with the chloroplast, and inside of the chloroplast is the chloroplast DNA. So we have to now separate the chloroplast from all the other junk that's in there. So we're using this centrifuge. There are many, many steps of centrifuge. I won't show you all of them because it gets kind of monotonous. But eventually, that little green pellet at the bottom, that is the chloroplast.
Hmm.
And so now we're breaking up the chloroplast and we're going to break it open and remove the DNA from inside of the chloroplast. So if we've cleaned it up, that's the chloroplast. We're going to cook it overnight. That's going to break the chloroplast open. You won't be able to see the DNA. The DNA is water soluble, so it just dissolves in water and it looks like water. Okay, so after they've been um, broken open overnight, that solution now contains the DNA. So now we have to purify the DNA. Again, this is a lengthy multi-step process. We separate the DNA from all the proteins. So in this vial, you'll see two layers. It'll The layer on the bottom is the protein, the layer on top is the DNA. So we're pulling that layer off the top. And then this is the first time in this whole process that you actually kind of get to see the DNA. You see that little thing floating around in there? There it is. Looks like a little white snowball.
Yeah.
That's the DNA from this whole process. So we've extracted the DNA from the cacti. This is the chloroplast DNA, not the nuclear DNA, and I can explain the difference here at the end. And then comes the fun step, sequencing. This was the step that I had never done before. In all my years in grad school, we just didn't have this technology back then. So a lot of different steps here, which I'm not explaining, and that's okay. We're mixing things with things, and that's that's how it works. Okay, now here comes the actual sequencing. So there's the min ion sequencer. Beautiful. And that clear solution is the DNA. We're adding it one drop at a time, and it's drawn into the sequencing device. And then here you can see as it's running, the number of reads, 26,000. The number of bases, 8 million bases. And this is what it looks like when it comes out. So each of those is a different DNA fragment that was threaded through one of those pores. We then take all those fragments and we line them up to assemble them into this finished sequence, which is a total of 149,000 base pairs long. It's in a circle because the chloroplast DNA is circular. And that is uh, the finished sequence from this whole process. A very lengthy process, but very rewarding to get to the end of it and see like what we can do now compared to just 10 years ago. And that, so let me pause there to say all of that stuff was funded by your donation. The actual sequencing device, the sequencer, the computer that analyzes the sequence, and that instrument for performing quality control on the sequence. So that none of this would have been possible without that donation.
Wow.
Great. And then we have about 5 more minutes here. Here you're
Okay, so then let me kind of cast the vision on where we're headed. I just showed you something from a living organism, the cactus, but we're really interested in trying to get DNA in long dead organisms, uh, such as the dinosaurs. That would be the holy grail of ancient DNA research if somebody could extract the sequence from a dinosaur fossil. Now, most of the time when people are digging for dinosaur bones, they don't care about the DNA because they don't think it exists in there. They so they put glue all over the fossils. They touch them with their hands. They do all that sort of thing. And we said that we want to try to get some clean, never touched fragments. And so we went to Glendale Dinosaur and Fossil Museum up in Montana. And this is a creation group, it's a really fantastic organization up there. And they took us out in the field and we went on a dig. And so what we're doing here is as soon as that bone was exposed, we got it. We separated it into a couple different fragments. These are very small bone fragments. They're fossils, fossilized. And we are going to be sending them to some different labs as well as our lab to try to see if we get any DNA or protein out of it. You can
Hey,
show
Don, just for going, I want to make sure that this is okay because I've got the screenshot. I don't have Joe up there. And does it matter as far as your video? I didn't mean to interrupt you like that, but I'm
no,
only
no,
seeing
no.
screenshots. I'm not seeing I'm not seeing Joe on the screen.
That's
I'm
okay.
hearing him.
Okay, we have then um, this one.
I love this picture for a couple reasons. So what we're doing, and this is different than, when you're out in the field as a paleontologist, you care about what the bones look like, not what's inside of them. And so uh, you typically don't store them in any special way. Here, we're using gloves to prevent contamination of the DNA, if there is any, and we don't know that there is. Uh, and then we vacuum sealed them. So that's what you see me doing there. We're running these through a vacuum sealer to remove all oxygen and moisture. And that's going to help preserve whatever small amounts of DNA are present. I like this picture because it's very realistic. Do you see my two kids in the background sitting out there in the heat along with us? Uh, that's my youngest, Genesis, and my oldest daughter, Alethea.
Okay.
Here's an example of a theropod tooth that we dug up and is one of the samples. We collected 10 samples. One of the samples that we will analyze for DNA. And this is what it looks like. I mean, we're just digging through. This is the Hell Creek Formation in Montana. It's Cretaceous in conventional terminology. This is a Cretaceous layer, and it has a lot of Edmontosaurus, duck-billed dinosaurs, as well as theropods. And so that's one of the samples that we were collecting. Just kind of give you a picture of what we're trying to do.
So have you begun the process then of trying to get DNA out yet or not?
Not yet. These are
Okay.
in the fridge. Actually, I brought some with me here. So
All right.
here's this one is a vertebra, a tail vertebra from an Edmontosaurus. It's just one of the fragments of it. We'll be sending the other pieces to other labs. So I have them here. We have our undergraduates arriving in a couple weeks, and this is going to be our fall
Oh,
research
this is
project.
exciting. That's
Yeah, I
really
can't.
exciting. You know, it really is.
Now
Well,
we have
we'll
to.
have to have you back on our show again to give us an update once you start getting some results from your experiments. But we're running out of time in our TV show. That's the trouble with a half-hour show. But this will be airing on several public access stations in Michigan and also on our YouTube channel, rae.org, under YouTube. So we hope that you enjoyed our show today. And thank you for joining us, Joel. We appreciate what you're doing and we'll keep up to date with you.
Thanks for having me.
Yeah, I enjoyed it. Very informative. Thank you.
We'll talk to you later and we'll see you next time on Revolution Against Evolution.